You said, I think we've heard quite a lot about how to make statistical inferences of proteins that are significantly changed due to some biological phenomenon in your proteomic experiment. And I think the question is now, once you have those lists of proteins, what do you do with them to sort of try and understand the pathways that they affect? And I think those questions are quite similar in the realm of proteomics and in the realm of genomics, because in genetics with next generation sequencing, you also sequence a number of genes in patient cohorts, and you get a number of genes that are significantly mutated, and you want to figure out whether or not they fall into a pathway. So this is why I think it's completely natural that I sort of fluctuate between the proteomic and genomic space, because in both areas we have these lists of genes or proteins that we want to analyze for functional connections to each other. And I'm going to give a very short introduction to web-based annotation tools used in the proteomics space. And this is basically going to be a resource list that you can then test and check out yourself. These different tools are quite well explained, so I think that the major reason for showing this is not to explain all of these tools to you, but just to make you aware of what the people in Steve Carr's group and other groups are using, and then you can start using these tools yourself because they're quite user-friendly. I'm then going to show you some more custom analyses that we've made on the tissue-specific post-translational modifications, and how we've tried to take the results from these tissue-specific PTM or post-translational modification experiments and map them to pathways, and then to try and understand what are the diseases affecting those pathways, and do they correlate with the tissue specificity of the PTMs that we use to start building the network. So those are some custom analyses that aren't really available from any tools that I can point you to, but we're hoping to make these sorts of custom analyses available to BroadE researchers and the BroadE community through the Spark mechanism, and through just making and talking to the bioinformatics platform about making new tools available to both the proteomics platform people, of course, but also the broader community at the BroadE Institute. So hopefully these custom tools will be available at some point in the future. So like I said, this is just a very boring resource list that it's there so you can have it in your handouts, so you can go through this list of resources. And I think there are a number of different resources we can use to annotate genetic and proteomic data. I'm not going to go through them, but I'm going to highlight 4 or 5 of these methods in the talk. And I think those should hopefully exemplify what are the sorts of tools out there, and then the rest of them you can just check out for yourself. And again, these are the tools that the proteomics people in Steve Carr's group use, so I think and also the people in the medical and population genetics here at the BroadE Institute. So I think they've been quite extensively vetted through those people, and I would recommend that you use any of these tools, and they're all very good, I think. So I've worked with sort of an in silico approach to mapping protein-protein interaction data, where we didn't get the data from proteomics experiments, but where we took data that was available in the public domain and mapped them to a human protein interaction network for genome annotation. And we've then integrated this protein interaction network into a tool that I'll show you in the next slide called Dapple, where you can actually analyze pathway relationships in this human protein interaction network. So basically, I just need to show you this slide so you can understand where the data that Dapple runs on, which is the next method I'm going to show you, where that data comes from. And this is a protein interaction network called InWeb. I'll be happy to distribute it to anybody who wants to use it. You can just send me an email. We're also gearing up to make a resource publication. But basically, we take all of the major protein-protein interaction databases existing in the public domain. We download and reformat the data. We transfer data from model organisms, from model organisms to humans. We have a very thorough cleaning up of all the data, because many of these protein-protein interactions are quite noisy. But of course, if you observe a given interaction between A and B multiple independent times across many different interaction publications in PubMed, you can then start to pile these observations on top of each other and get a confidence score on the individual interactions. And what we end up with is a human protein interaction network. It has approximately 500,000 interactions now, between 12,500 proteins. And I think the important thing is that for every one of these edges or interactions, we have a confidence score between 0 and 1, which reflects how much we trust the data. And we also can trace back the exact origin of every interaction. So I can tell you exactly which database does it come from, which experimental technique and which publication. So you would basically, if you find an interesting interaction in InWeb that somehow supports your data or it generates a hypothesis in your head, then you could go back and read the source publication, so you could actually be sure that they did the experiment properly and that it isn't an annotation mistake or anything of that sort. And I think that's those are two key elements of InWeb, the ability to trace back protein interaction data, and that we have a uniform and quantitative protein confidence score in each individual edge. And this is something that Dapple, which stands for Disease-Associated Protein-Protein Link Evaluator, utilizes. So Dapple is an algorithm that runs on InWeb, and it was actually built to study genome-wide association data, but basically can take any gene list that you throw into it and figure out if there's a statistical significant amount of connections between these proteins. So I'm sure that many of us have seen networks generated from proteomic or genetic data where you really don't know if the networks that you're seeing have any statistical significance or if it's just spurious connections that you're seeing between genes. And this is a method that can really tell you this. And like I said, whether the input set comes from a proteomic experiment or from a genomic experiment, it doesn't really matter. But in this case, the input protein set came from a genomic data set of genome-wide association studies in Crohn's disease. And those studies all pointed to a number of proteins colored or exemplified here by these large colored nodes. And we then connected through protein interaction data in InWeb and through the Dapple algorithm, connected these into a network and quantified the amount of connections between these proteins. Yeah, I'm sorry, there's something about the air in here, but thanks. So Dapple runs through a statistical test of the connectivity of these proteins, and it takes into consideration the exact architecture of InWeb. So it's really a non-confounded test based on the null distribution of random InWebs, where we've completely permuted the InWeb protein interaction network while keeping the genetic or the architecture of the interaction network intact. So you basically start to assemble these proteins into networks and pathways like this. And I think what really got our attention was that some of the proteins that are pulled into this network by the seed proteins actually turned out to be involved in Crohn's disease in subsequent follow-up genetic tests. So basically, not only could we start to make a passive annotation of the data and assemble it into a pathway, we could also start to predict things that weren't even published at the time, and we could actually start to predict new genetic findings that came out subsequently. So I think that was a key element and really an early success for Dapple. And I should say, I forgot to mention this specifically, I think it's probably obvious from this figure, but Lizzie Rosen developed Dapple, and she's been a fantastic collaborator, and it was developed in the lab of Mark Daly. So it turns out that Dapple has been an extremely flexible resource. It's recently been applied to exome sequencing data in autism, but again, this is just a gene list that was then connected into these networks, most importantly a network in chromatin remodeling, and it wasn't in any way known that chromatin remodeling would be an important factor in autism. So the fact that we could take exome sequencing data from patients in autism and connect them and find a chromatin remodeling network that was statistically very significant was a key finding in this paper. And I think it was especially interesting because it was replicated in another paper from Brian O'Rourke and Evan Eichler that's basically using another patient cohort also find a chromatin remodeling network that looked a lot like this one. So we feel that this is a pretty, even though we haven't followed up on this study, we could certainly do that using proteomic technologies, but even though we haven't followed up on it, I think the fact that another group in the same, these papers came out back to back in Nature, they found a similar network that was really a key interest. Again, this method has now been used in a number, also in type 2 diabetes and inflammatory bowel diseases, and my point is just that again and again and again we see that if you run these Dapple networks on gene lists or protein lists, you can start to make networks that were unexpected, and these networks have predictive power because they have been able to predict new genes involved in type 2 diabetes or new genes involved in inflammatory bowel diseases. So I think you're getting more than you bargained for maybe. You're not just connecting your gene sets into a pathway, you're actually pulling in new things that you can then follow up in a hypothesis-driven manner. And I think that's a key message from the Dapple. And Dapple is available from the BroadE Institute website. There are, of course, many other tools. One example is the tool David, which is a very flexible bioinformatics tool where you can input gene lists, and then you have a number of analytical modules you can implement. You can make functional annotation, you can make different types of charts, you can make gene functional classifications, and you can sort of check these different analytical modules, and you get a number of results lists that you can also integrate in a very nice and uniform manner. And these give you gene ontology, enrichment, categories, and so forth. And this turns out to be a really flexible and nice tool for annotating proteomics data sets. And it's a really well-described resource too. So I would, if you think this is of interest to you, you should read. There is a really nice Nature Protocols paper, and also some very thorough descriptions of this method on its website. And also, again, this has been vetted because it's extensively used in the proteomics communities that are, or in the, I would say, the top proteomics groups. Another tool is developed by Rune Linding and Lars Juhl Jensen. It's called NetworkKin. And this is a tool where you can take and map kinases, if you've been studying kinases, to substrates. So the problem with kinases and substrates is that if you use sequence-based methods, kinases can, in principle, phosphorylate many different proteins, but in vivo they actually don't phosphorylate many proteins because they don't have access to those proteins in the tissue in question. So what NetworkKin does is it doesn't only use sequence motifs, it also uses the biological network contexts through the string database to assign not only which kinase can phosphorylate which substrate, but which kinase can phosphorylate which substrate, and which kinase would actually meet that substrate in a biological context. So those are, this cleans up these predictions quite a lot and makes this a very powerful tool for that type of analysis. And there's a new version of this coming out on a yearly basis, I think. So a very nice tool. Of course, I'm sure that many of you also know the string database. This is also an excellent database. In these string networks, these nodes represent proteins, and these edges represent functional associations between the proteins, which can be text mining relationships between the proteins. It can be that the genes are co-regulated across tissues. It could be that they have a genomic context that suggests that they're functionally associated, and by genomic context, the string people mean that they are present in the same genomes and absent in the same genomes, which would indicate that they're functionally associated to each other because they always pop up as pairs. I think they're testing 6,000 genomes. So one of the major drawbacks, I think, of the string database until the new database that just came out was that it didn't have a statistical test on these networks. But I just saw the paper came out, I think, 2 days ago, and in the new version of string, they actually implement a test, much like Dapple, where you throw in your set of proteins, and then strings figures out, is there a significant amount of connectivity between your proteins? So I think that was a key element that was really needed in this type of analysis, and it seems like that's up and running now. So again, you may consider it as an advantage or a drawback that these are not protein-protein interactions. I think that depends completely on what you want to do. Of course, you have, I would presume that there is more coverage in these data types because you can draw on many complementary functional association data. But I would say a challenge is to actually interpret. So if you see this kind of network and half of it is based on gene expression correlations, the other half on genomic context data, I wouldn't know specifically which experiment to carry out to functionally test this network. In contrast, if you have a protein interaction network and you want to confirm the specific interactions that you're seeing, I would know which experiment to carry out because I would do a protein-protein interaction follow-up analysis. So I think those are the things that you need to balance a little bit. But if you just want to annotate your data, I think this is another excellent resource for that. So I think this was a whirlwind tour of the different tools that you can try out yourself, and there were other tools available from the list that's in the handout material. And now I'm going to proceed to what I would call some more interesting and some more advanced analyses of post-translational modification data. So one thing we of course want to do when we have a set of proteins is to reverse engineer them into molecular networks. And the way that we've typically done this is to follow this scheme, that if you have a set of proteins, we call them seed proteins. You then test in InWeb what do they connect to, and you start to pull in significant linker proteins, which means proteins that significantly connect to your input set, and you can then start to annotate these full networks. And you can do pathway analyses either manually or in a semi-automated step, and start to see that maybe these clusters correspond to specific pathways. And then again, you want to do a statistical test of your network to see that these connections that you're seeing are not just spurious, but they actually have some biological meaning and are beyond being what you would expect by random connections between the proteins. So in the case where you want to do that, you have to define some network score. You can come up with many different network scores. You can measure the connectivity of proteins. You can come up with other metrics for this type of network analysis. But in this case, we'll just consider a very simple network score that has turned out to work quite well empirically, which is the amount of seed proteins in the total out of the total amount of proteins in this network. In this case, there are 54 orange seed proteins that we seeded the network on, and there are 174 proteins in total, so the network score would be 0.31. And the question is now, how likely is that how likely is it that we'll observe that by random? And we basically do permutation tests, because these protein network analyses, we really don't have distributions that we can test this against, so we have we actually have to calculate the distributions for this specific set of proteins, and we do that by throwing in random sets of proteins that are matched completely to the seed set, and then we can actually make the distribution of network scores, and we can simply read out the p-value. And in this case, this is a very significant network. We would never expect to see this amount of seed proteins in the total amount of, in the full network by random. So the p-value in this case is 1.7 times 10 to the minus 5, and this is actually a hard developmental network, but it's just used here for illustrative purposes. But this is an approach we use to annotate and analyze two data sets of post-translational modifications in a collaboration with the Jesper Olsen's group in Denmark. And basically they had analyzed tissue-specific acetylation sites across 14 tissues. And the question was, okay, if you have a set of acetylation sites that are seen in brain, do the proteins that have a brain-specific acetylation pattern actually, do they form brain-specific pathways? I think that's the main question. And also, if they do form brain-specific pathways, are those pathways then related to brain-specific diseases? That was what we wanted to know. So what we basically used was the data set of acetylation sites mapped to 16 tissues. For every one of these 16 tissues, we made a tissue-specific network following the strategy that I just showed you. We mapped OMIM diseases, which is the online Mendelian inheritance in man database, onto those networks and measured which diseases are significantly represented by proteins and genes in the network. And then the example that I'm going to show you now is a network based on brain-specific acetylation. And like I said, we took proteins that were found to be acetylated in the brain. We expanded the network using the method that I just showed you. And then we mapped onto this network the pathways that emerge, and it turned out that these pathways are clearly brain-specific. I mean, it's not perfect. It's not like we're getting only brain-specific pathways. We also get some clusters of unknown function that we don't really know. We get some function, some clusters that have ambiguous functions, but clearly overall this is what you would consider to be brain-relevant biology. And I think that's even more supported that when we map the OMIM diseases onto the network, so we basically asked, if we take all of the proteins in this brain-specific network and find the corresponding genes, is this gene set enriched for any disease in OMIM, and if so, which disease? And in this case, there was only one disease that was specifically enriched in this network, and that was familial epilepsy. So basically, epilepsy, two of the genes in this cluster, in this voltage-gated calcium channel cluster up here, that have a brain-specific acetylation on one of the proteins, are mutated in familial epilepsy. So I think that was a really nice example of how you can start to integrate these PTMs that have a tissue-specific signal into tissue-specific pathways that also reflect tissue-specific diseases. We did a similar thing for another paper that Jesper Olsen's group also produced. This was phosphorylation data, and the phosphorylation data was I don't know if of higher quality is the correct word, but it was easier to map the tissue-specific phosphorylation sites. So we got, I think, even better results for that. Again, Alicia Lundby and Jesper Olsen, they mapped phosphorylation sites across 14 tissues. We took these 14 different data sets and made tissue-specific networks using the method that I showed you a few slides ago. Again, we mapped diseases onto the network. And in the example that I'm going to show you is a network based on blood-specific phosphorylation. So again, the green proteins are those that have a blood-specific phosphorylation site. And again, this network clearly has blood-specific biology. It's even more clear than the brain network. We have blood coagulation, inflammation processes. There's also a cluster of unknown function that didn't really have a strong uniform annotation. Immune regulation, anion exchange complex, erythrocyte membrane complex, and other things that are clearly relevant to to blood. And again, the only disease that was significantly enriched in this network was a disease of bleeding. So it's something called Mendelian congenital aphibrogenemia, and 3 proteins in the blood coagulation cluster. Some of actually 2 of 3 proteins have a blood-specific phosphorylation site. They're involved in, known to be muted in families that have these disorders of aphibrogenemia. So again, the tissue-specific phosphorylations clearly define tissue-specific pathways that are involved in tissue-specific diseases, and I think that's, of course, what we're hoping to find when we map these tissue-specific post-translational modification sites. So unfortunately, these tools are not available, but we're hoping to make them available. I'm going to show you that in a few slides. Just another example of how we can reverse engineer networks based on lists of proteins. In this case, we had a number of proteins. All the orange proteins are known to be involved in aortic valve development. I'm very interested in the cardiovascular disorders, so we set out to see, can we map an aortic valve developmental network? So we basically ran through the algorithm that I showed you again a few slides ago, and this network popped up, and this was an extremely significant network, and we were able to prioritize genes within the network that were not known to be involved in aortic valve development based on their connectivity profile to the proteins that are known to be involved in aortic valve development. And 3 or 4 proteins came up with a very significant score. They're ranked here. So if the red protein up here is the most significantly connected to the orange proteins, the dark green one is the next most, and the light green one is significant but less so. And one of the proteins that popped up was SNX9, and we specifically chose to validate that one because it was completely unknown to be involved in any type of heart developmental biology. So we really specifically targeted those proteins where no one could say after the fact that we knew a little bit already that they were involved in heart development. So if there was just any minor piece of evidence connecting these proteins to the heart, we actually chose not to validate them. We chose the ones that were completely novel, and for that reason we chose SNX9. We did immunohistochemistry in a 6-week-old aborted fetus that wasn't aborted for heart reasons, but it was a natural abortion. For other reasons, and it turns out that SNX9 is actually specifically expressed in the leading edge of these are called endocardial cushions, and this is the anatomical precursor to the heart valve, so that strongly suggests, of course, it's not a functional validation of SNX9, but we tested 6 other time points in heart development and 6 other anatomical structures, and SNX9 was not expressed in any other structure at any other time point, except for the valves. Again, it was actually also expressed in later stages of aortic valve development, again in the leading edges of the valve that's sort of growing. So I wouldn't call this a complete proof that SNX9 is involved in aortic valve development, but it's strong supportive evidence. And in fact, we didn't just make a heart valve network, we made 19 different networks of 19 different anatomical structures in heart development. We made one for the atrial septum, one from the endocardial cushions that I just showed you, the outflow tract, the myocardial trabeculae, and for 15 other structures. And for all these networks, we ran through the procedure that I just showed you. We picked out proteins that were predicted in the network to be significantly interacting with the seed set. And we then validated those proteins using immunohistochemistry across 19 human hearts, across 6 developmental time points. And we actually had, so we had a total of 382 tissue sections where the same guy using the same microscope, he, in a blinded way, sat down and scored the expression patterns of these different proteins that we tested. And he didn't know where we expected them to be or what proteins he were looking at, so he had no idea. He simply sat down and annotated where are they expressed, and we then correlated the predictions of the networks to the expression profiles of these proteins, and we saw that in 72% of the cases, the proteins are actually expressed specifically where we would predict them to be expressed based on their connectivity into these networks. So I think this is an example of how you can take lists of proteins, you can devise a developmental network that has predictive capabilities, and you can then follow up that those predictions using, well, that depends on the biology that you're studying, but in this case, immunohistochemistry. We also did a lot of gene expression analyses that further confirmed both the proteins that I'm showing you here, but also a larger set of proteins predicted by the networks. So just one slide on how we can integrate these data with genetic data. I think that probably also has broad interest. One way that we're, of course, thinking of taking these networks and then integrating with genetic data, and we could do this with other proteomic networks that we're discovering through proteomics and sort of in silico analyses of the network relationships, is that we want to filter exomic variants, that we have access to 174 families where you have 2 healthy parents, but 1 child who has a bicuspid aortic valve, so an aortic valve defect. And of course, we want to figure out, do any of these kids have a de novo mutation in any of the genes that are represented in this network? Because we believe that this is an aortic valve developmental network, and then the hypothesis would be that maybe de novo mutations in these kids in any of the proteins in this network are actually driving that abnormal valve development. So you could, of course, do this for you could also integrate these types of networks with genome-wide association data or cancer sequencing data. But this is just an example of how you can sort of go between the spheres of proteomics and genetics using these bioinformatics network building tools. This is something that we hope to get up and running soon, basically. My talk is not the longest of the talks. It's actually, there's only a few slides left. So I guess that might be good timing since I'm also the last person. But just to give you an idea of emerging tools, because of course we want to make these things available to people. We don't just want me to be able to run on my computer these types of analysis. We want to enable BroadE and the BroadE community and collaborators to be able to do this. And fortunately, the BroadE Institute and specifically the Spark Committee agreed that this would be a good idea, so they just funded me and Daniel MacArthur and Aviv Regev and Jill Mesirov to make a web portal where we'll try to make all of these biological networks available. We'll make InWeb available, String, and other networks. We'll try to, or we will build a set of tools that can analyze and integrate these different networks. So you basically will be able to go to this portal and throw in your sets of proteins, and this portal will then build you the network that emerges from your set of proteins. And hopefully, like I've shown you in some of these projects, that will give you a functional interpretation of your proteomics data or your genetic data for that matter. And that will hopefully also suggest targeted follow-up experiments. So just to give you one example of how we've used this, in arrhythmias, we had a set of proteins that through genetic analyses were incriminated in arrhythmia. We made a network using InWeb and Dapple. And it was pretty obvious when we looked at that network that if we were to carry out specific proteomics experiments of specific cardiac ion channels, we would probably get a very good signal, and that we would probably also be able to identify new players involved in cardiac repolarization, which is the biological mechanism underlying arrhythmias. And it turned out that when we did targeted proteomic follow-up experiments, exactly as suggested by this bioinformatics analysis, we did see that signal, and we were able to predict new proteins and new genes involved in cardiac repolarization and arrhythmias. So I think we don't consider these networks to be an end game. In some cases, you just want an annotation of your data, but in some cases, you want the annotation to actually drive a hypothesis that you can then test, and we consider these networks as a way to make really targeted follow-up experiments, because you already have, if you do get a signal in these pilot analyses, you actually have already a pretty good indication that if you were to carry out specific experiments, you would probably see a positive result. So this is, we kind of see this as a circle. This isn't an endpoint for us. This is the beginning of a specific proteomic experiment or a specific genetic experiment. So So that's how we hope that other people will also use the resource. Not that there's anything wrong with annotating the data, but I think it's more interesting if we can use these in silico networks to really drive biological experimental validation. Again, just the resource list, I don't know why I have it twice. I guess it's just to remind you that these are the resources that are being used in the community. And then I would, of course, like to thank a lot of people. As you can imagine, many of these projects are very collaborative. And I apologize again for my cough. I don't know what it is with the air in here, but something happened the minute I stepped down here. So, but you can read the people who are involved. But I particularly want to thank Mark Daly, Lizzie Rossen for their contribution, or actually driving the Dapple project. And also, of course, I want to thank very much Steve Carr and the Proteomics group here for inviting me to give a talk. And thank you very much for listening. I hope that you can use the handouts to sort of implement these methods yourselves. So thank you very much.
Let's thank Casper. InWeb or Dapple allow you to filter the large interaction network into, or let the interactions that you are interested in be dictated by the expression of the genes. So filtering out edges where the partners may not be expressed in the same tissue or something.
Yeah, so the question was, can you use, for example, tissue-specific expression data to filter out edges from InWeb and then make a more tissue-specific protein interaction network? Is that correct? Yeah, and I think that's a great idea. We're actually hoping to partition InWeb. So right now, InWeb is a completely non-cell type specific, non-tissue specific network. And I think what we're hoping to do is two things. We're hoping to, one, partition InWeb into tissue-specific subsets, where we can maybe come up with tissue-specific ways of optimizing the network, so you get better data for a heart network and a brain network and so forth. And I think the second thing that we're really excited about is all these new methods that are coming out of Steve Carr's proteomics group to make draft molecular networks pretty quickly and pretty cheaply. Because why not just run all our algorithms on custom protein-protein interaction data from the proteomics platform? That would be much better if we can generate those data cheaply and with good resolution, right? So all the algorithms we're building on in silico data, the only reason why we build them on in silico data is because we don't have access to actual high-quality tissue-specific protein interactions. If we had that, I think maybe InWeb will not be obsolete because we can use it for other things, but I could imagine a scenario where we have such good data sets from different tissues that we don't, we waste, or we spend quite a lot of energy on updating InWeb, and maybe in the future that won't be necessary. So. All right, thank you.